All right, everyone. Today, we are diving into a really exciting new chapter in the gene editing revolution. Now, you've definitely heard of the rock star CRISPR-Cas9, right? We'll get ready to meet its incredibly versatile and powerful sibling, a tool that's changing not just how we edit our genes, but how we detect disease itself. Think about that for a second. We're talking about a single technology that both opens up completely new parts of the genome for editing and gives us a lightning-fast way to diagnose diseases. So this isn't about replacing Cas9, not at all. It's about a massive expansion of the entire genetic toolkit we have to work with. Okay, so let's get into it. For years, Cas9 has been the big name, right? The face of CRISPR technology, the molecular scissors that really made precision gene editing a household idea. But, science doesn't just stop there. So meet Cas12a, which you might also hear called CPF1. It's in the same family, for sure, but it comes with some truly unique skills that really set it apart. Now, while Cas12a is a fantastic gene editor, its first big claim to fame actually comes from a kind of secret superpower, one that Cas9 completely lacks. And this ability has turned it into a revolutionary tool for diagnostics. You can think of it like a molecular sentry that can spot an intruder, like a virus, with just incredible speed and accuracy. So what is this superpower? Well, it's this really cool phenomenon called collateral cleavage. Here's how it works. Cas12a, with its guide RNA, finds its specific DNA target. Let's say it's a piece of viral DNA. It makes its cut just like you'd expect. But then, and this is the wild part, the act of cutting that primary target flips a switch. It activates the Cas12a enzyme. It's exactly like a security guard who, after catching the one specific intruder they were looking for, suddenly starts shredding every single piece of paper in the room as a giant alarm. So, of course, scientists figured out a really clever way to use this shredding behavior in a system they call detector. First, they take a sample and make a ton of copies of any target DNA that might be in there. Then, they add the Cas12a complex and these special little reporter molecules. These are just short DNA strands with a tiny fluorescent light switch that's normally turned off. If Cas12a finds its target, boom, it gets activated and just starts shredding everything in sight, including those reporter molecules. And the result? It's just beautiful in its simplicity. When those reporter molecules get shredded, their little light switch gets flipped on, and you get this fluorescent glow. So if the sample glows, yep, the target DNA is there. If it doesn't, nope, it's not. It is a super clear, definitive yes or no answer, and it happens in just minutes. I mean, this technology completely turns diagnostics on its head. It's incredibly sensitive, it can detect even tiny amounts of a pathogen. It's fast, it's simple, and it doesn't need a bunch of complicated lab equipment. And that opens the door for true point-of-care testing. Just imagine getting a rapid, highly accurate diagnosis for a virus or even a cancer marker, right there in a doctor's office or a local clinic. But as revolutionary as Cas12a is for diagnostics, and it really is, that's only half the story. It is also a formidable gene editor in its own right. And in this role, its unique properties give it a distinct edge, letting it tackle genetic problems that are really challenging for its more famous cousin, Cas9. So this leads to the big question, right? If you've already got Cas9, why do you even need Cas12a? Well, it turns out that having both in your toolbox dramatically expands what's possible. They're not really competitors. Think of them more like partners. All right, let's just break down the key differences side by side. First off, size. Cas12a is smaller. Its guide RNA is also simpler, which makes it a bit easier to work with. But probably the most critical difference is this thing called the PAM sequence. It's basically the little address label the enzyme needs to find its target. Cas9 looks for a G-rich one, while Cas12a looks for a T-rich one. And finally, the way it cuts. Cas12a makes a staggered cut, leaving what we call sticky ends, which can be a huge advantage for inserting new DNA. So what does all that technical stuff mean in the real world? Well, the smaller size, that is a huge deal for actually delivering the editor into a patient's cells. And that different PAM sequence? Oh man, that is a massive game changer. It means Cas12a can target parts of the genome that are completely invisible to Cas9, effectively expanding the map of what we can edit. The simpler guide makes research faster, and those sticky ends we mentioned, they can make it way easier to precisely insert a new correct piece of DNA. So all of these advantages, they're not just interesting facts for a textbook. They are actively pushing the boundaries of what we thought was possible in gene therapy, and they're opening up brand new avenues for treating some really devastating genetic diseases. Let's really zoom in on that size difference for a moment. Being about 20% smaller than Cas9 is a massive advantage. You see, the most common delivery vehicle for gene therapy inside the body is a harmless virus called an AAV, an adeno-associated virus. And the problem is it has very limited cargo space.
And this brings us to a really critical point. Fitting the entire gene editing system into that AAV delivery truck is a major bottleneck in the field. Cas12a's smaller size just makes it a much better fit. This simplifies the whole process for in vivo therapies, that is, treatments given directly into the body to reach organs like the eyes, muscles, or the liver.
This directly translates into real hope for specific diseases. Think about hereditary blindness, where you can deliver Cas12a right to the eye. Or something like Duchenne muscular dystrophy, where you have to reach muscle cells all over the body. And crucially, it lets us go after genetic mutations in parts of the genome where Cas9 simply can't land because its required address label just isn't there. Cas12a unlocks these previously untreated targets. So when you step back and look at the big picture, the arrival of tools like Cas12a represents a fundamental shift. It's not just an alternative to Cas9. It's a complementary partner that expands what we can do in both diagnostics and therapeutics. It pushes us from an era of possibility into an era of application, and it forces us to confront a brand new and, honestly, a much better kind of question.